Okay, so we're talking about PCR. And essentially, PCR is a mechanism that allows for DNA replication to happen in a test tube. So you have a double-stranded DNA, and you need to separate the strands. The only way to separate them is to heat them at a high temperature. And high means maybe 90 degrees centigrade or so. That is a high enough temperature to break the hydrogen bonds and make the two DNA strands separate. And then you have to keep them separated. In a live cell, what keeps them separated are helicases. And then stabilizing proteins will keep the two strands apart. Well, you cannot do that in a test tube. That would be too cumbersome. It's not practical. It cannot be done. However, there's something else that could be done. Lower the temperature just enough to keep the strands separated. So you might lower it to 75 degrees centigrade, which is still very, very hot. They're still separated, but it's not 90 degrees. Now it's a little bit less. And then this is the clever part. What he thought of is to use a DNA polymerase from a thermophile, from a thermophilic bacteria. A thermophilic bacteria is one that only grows at high temperatures, which means its enzymes can only work at high temperatures. And so, as opposed to all other enzymes, which will be the nature at high temperatures, these enzymes not only survive, but those are the only temperatures at which they work. So he used the DNA polymerase from a thermophile. He used a primer, which he made, just a short piece of RNA. And let's go back here. Again, if you want to replicate a DNA, you have to know something about that DNA. You have to know at least a few of the nucleotide sequences to be able to create a primer. And then from the primer, he would add the DNA polymerase from the thermophile, which will continue on the replication, and it will make copies of the DNA. And then he will go through the whole cycle again, separating the pieces. But now, instead of just having 2 templates, now you would have 4. And then on the next cycle, you would have 8 templates, and then the next one, 16. And this way, within a matter of hours, instead of weeks, you would have a large number of copies of the DNA that you desire. So that's what PCR did. It created a very, is this the PCR in syphilis PCR test? Yes, absolutely. Yeah, we use PCR nowadays to identify many bacteria. I mean, that's the same PCR that they're using to identify COVID. They use what's called reverse PCR because now we're replicating RNA, not DNA. But the idea is the same. And it is, yes, it is the same PCR that is used for the blood test. Yeah. So, but again, it's essentially taking the patient's sample, bursting the cells, getting whatever DNA is in there, and then we should know something about the sequence of the syphilis bacteria DNA. Maybe we know a little piece of it so that we can make a primer. So the primer is put into the test tube, DNA polymerase is put into the test tube, and now the key here is the primer. So in this case, the primer is what acts as a probe. If the patient has syphilis, then the primer will be able to bind to some of the DNA in the patient's sample because some of the DNA is coming from the syphilis bacteria. If the patient doesn't have, and then PCR will be able to replicate that bacterial DNA. If the patient doesn't have syphilis, the primer will not be able to bind to anything, and there will be no DNA replication. That's basically what the way it works. How much DNA you get within a certain amount of time can tell you how much DNA of the bacteria is present in the sample. The more DNA of the bacteria present in the sample, the more DNA will be made through PCR because there's tons of templates. So if the patient has a very, very high infection, there will be lots of the bacterial DNA that can be used as templates to copy. And so you would get, within a matter of a few hours or a few cycles, you're going to get tons of the bacterial DNA that has been made. If the patient has a small load, there might only be a few less DNA from the bacteria, and therefore, through the same amount of time, less DNA will be made. There's less templates to copy. So that's the idea of PCR. So nowadays, initially it was just a way to replicate DNA. Nowadays, it is not only a way to replicate DNA, it's a way to replicate the DNA and identify the presence of the DNA from a specific pathogen. So that's basically how that works. Yeah, it is very neat. Absolutely. Absolutely. And it's very useful because prior to these, it was very difficult to test for syphilis. It would be, there was an antigen antibody test, which was not very specific. That was a screening test, which is still probably is used. And then after that, you would have to, the RPR, there you go, RPR test. Correct. It's not very specific at all, but it's a good screening. It's cheap. It's a good screening test. And then from there, you would take it to a more, you would have to, you couldn't grow the bacteria, so there was no growing. Some type of blood, perhaps, which are expensive. Fluorescent antibodies of test, which again could be expensive and time consuming. This is very quick. This is very, very quick. And that's the other advantage. So, yeah, so what the Lab 9 actually focuses on is the DNA fingerprinting and DNA profiling, which we'll talk about here in just a second, which is also used to identify organisms. So essentially what's going to happen in this test is that restriction enzymes are going to be used to cut DNA. And if we have a bacterial DNA that has been it's sequenced, and by the way, a lot, if not most, of the most common bacterial pathogens have been sequenced by now. So we do have the sequence of most of the most important pathogens out there. So we have a bacterial DNA that, and we know the sequence. So we have several samples of the bacterial DNA. We know the sequence, so we know when certain restriction enzymes are applied to that DNA. And let's just say 2 or 3 of these restriction enzymes. We know the pieces that we get. So let's say we use those 2 restriction enzymes, we're going to get 1 large piece and 1 small and 1 tiny piece. Okay, so that's the cut that is being made. So actually, let me introduce 2 cuts for the blue one, and let's make it, let me think here. So if I cut here, then I'm going to have this cut right here. I'm going to have this piece right here, and then I'm going to have this piece right here. So I have a big piece, I have a medium piece, and I have a tiny piece. So I know the pattern I get. So the next question is, if I have this patient that I suspect may have this particular bacteria, and I take its DNA or a sample, and then I begin cutting and separating, is there in any of those, that DNA that I have, this particular sample that I'm looking at? So this particular pattern, I should say, that I'm looking for. So let me see. Okay, so here you have 2, as an example, 2 nucleotide sequences from the DNA of 2 different bacteria. And we're only going to worry about which prime the sequence is on. Okay. When you say which prime the sequence is on, what do you mean by prime? I'm not quite under, which is, oh, I see what you're saying. Okay. Actually, because the sequences are complementary, it will be found on both strands. So, and I didn't want to go into that here because I didn't want to complicate it, but yeah, you do. You will find it on both strands. Okay, so let's have this one. I'm going to stop. Okay. I'm going to stop right there. I think it doesn't matter. I think I can show you either way. Okay, so let me make the other strand. The other piece will be C, G, T, A, T, A. I don't know what that is. Oh, I guess that's an A. Let's make a T, G. Let's make this one a better A. Okay, so let's say that, give me a second. I did something wrong here. Okay. Give me a second. Why it's not working out for me? G T A T A T G. Okay. Okay, that's fine. Yeah, it works out. Let's say that the sequence recognizes a cuts every time there is a CG. That's sorry guys, this is not working out for me. Okay, this is a good one. AT, and it's going to cut, let's say right here, it would have another AT right there. It will cut right there too. So, and I'm gonna rephrase what I said. Some enzymes will cut on both sides, some will not. And some enzymes, when they cut on both sides, they make what are called blunt ends. So that what they cut ends up like these. Okay, so they cut like that, and you have same clear straight cuts on both ends. Other enzymes, when they cut both strands, they make what they call a sticky ends. And a sticky ends means that it's gonna cut like that. So we'll cut a sequence like that, and this is what we're gonna get at the end. So the two pieces that have been cut, both of them are going to have little pieces of single-stranded DNA right there. So to go back to your question, yes, restriction enzymes oftentimes, or most of the time, are going to code both, are going to have a long enough sequence that they recognize, and the sequence will be found, or I should say a short enough sequence on both strands. A short sequence will be found on both strands, and it will code on both strands. So they're going to cut on both strands, not on just one. This example, just for simplifying things, I only gave you one strand. So in this case, one of the enzymes cuts every time there is a CAT, and it cuts between the C and the A. So in this particular sequence, you will end up with a cut here and a cut here. And the second example, the second bacteria end up with two cuts. So when you run the gel for the first bacteria, you're going to end up with two pieces that are 1, 2, 3, 4, 5, 1, 2. Yeah, two pieces that are very similar in size, and one that is larger. You'll have the larger piece at the top, and then the other two, which are very close together because they're very similar in size. Whereas bacteria number 2, you end up with a huge piece, very large piece, which will not travel very far. That will be the first bar, and the other one will be very tiny, so it's going to travel very, very far down here. Okay. So the pattern that you get when you cut with the same restriction enzymes, these different bacteria can allow you to identify the bacteria. So that's the idea behind this DNA fingerprinting or DNA profiling. So you have the pattern for bacteria 1, the pattern for bacteria 2, and this is the unknown bacteria. You run the unknown bacteria, and you compare the pattern of the bars, and in this particular example, they matched bacteria number 1. I mean, they could have matched neither, in which case you didn't have 1 or 2. So that's essentially how this is going to be done. Okay. And that's what the exercise is going to do for you. That's what they're going to ask you to do. By the way, just as a reminder, remember that this works because the agar acts like a sieve. The larger the DNA, the harder the DNA it is for the DNA to travel through those pores, because it's like a huge snake trying to weave its way through tiny little pores. So it's going to take longer. If the piece is tiny, then it goes very quickly through the gel. So the smaller the piece, the further it travel through the gel. So this is a much smaller piece than this one right here. So just remember that this will be smaller than that one. Okay. No, there's not a difference between agar and agarose. So an agarose gel is an agar gel. Yeah, there's no difference. So you can have different concentrations of agar, but an agarose gel is essentially the same. It's the same agar that we use for the plates, for the bitter plates. It's pretty much the same thing. In the probes, yeah. So yeah, we talked about that. We talked about the probes, even though we don't really do the probes in this lab. Okay, that I can remember. I don't think we did probes. Okay, so we cut the DNA with restrictions enzymes. Okay, just looking at that, and digest, and incubate it for a time. Yeah, you incubate it for about 30 minutes to an hour to let the enzyme work. Yeah, the reason why we always buffer is because we're dealing with enzymes. And remember, enzymes require specific environments, specific salt concentration and pH, and that's what the buffer does. It gives the specific environment the enzyme needs to work. It's incubated because, again, these are enzymes from mesophiles. So mesophiles like 35, 37 degrees centigrade, so their enzymes will work best at those temperatures. You put prepare electrophoresis gel. The loading dye is from thymol blue and glycerol, so that gives a little bit of a color to the DNA, so we can see it running through the gel. The glycerol is going to weigh down the gel it's going to weigh down the DNA so that it goes into the well. Otherwise, it'll float into the buffer. This is the first time we use a ladder. This is again something we buy, and it has pieces of DNA of known size. It's a matter of comparing how far the ladder travels with your bars, and you have an idea of how the size of the sequences. And in this exercise, so does the glycerol bind with the DNA? Is that what allows it to weigh down, or does it just? No, it actually binds with the DNA. Glycerol is an alcohol, so it would bind with the DNA, and without distorting it, without damaging it, because ultimately it's more like a solvent, but it's a heavier solvent than water. The buffer is essentially water, mostly water. So when you dissolve the DNA in something that's a little bit heavier than water, then it will be more dense and it will go down with it. The DNA will go down with the glycerol. So that's the idea behind it. In this exercise, we have the DNA from Salmonella, and there are different strains of Salmonella. So there's a particular strain that had been causing an outbreak in a location. So they have 5 patients who have been testing positive for Salmonella. They have a Salmonella infection, but they don't know whether it's the same Salmonella that is causing the outbreak or a different Salmonella. And so that's what they're running the gel. They take the patients that have been infected and they isolate the Salmonella from the patients. They take the DNA from the Salmonella from each of the 5 patients and they cut this Salmonella DNA with 2 restriction enzymes, E. coli 1 and PST1. And then they're going to run them through gels and compare the pattern of the bands from the patient's Salmonella DNA versus the known. So the control is going to be the Salmonella strain that has been causing the outbreak. Okay. So that's what we're going to compare with. Okay. Any questions about these? Correct. We don't need a negative control because it does either matches or it doesn't match. Exactly. Yes. All we need is a positive control, which is something to match it with, essentially. Yeah, that's all we need. That's correct. I do have the lab report also. Yeah, go ahead. What's the question about the ladder? Okay, give me a second.
My question is, one of the questions asked.
Go ahead.
One of the questions it asks the approximate size of the largest fragment. And
okay.
Compare it to like the largest fragment was under the third rung of the ladder, but they weren't evenly distributed. It wasn't like
so
like what estimate, or do we do like it's smaller than 4,361 base pairs, or?
Right. You're going to have to approximate.
Okay.
So typically what we do, typically what it's done, we cannot do this, in this case because we're just working with a picture. But typically what it's done is we have the gel and we actually measure with a ruler how far the fragments on the ladder are from the well. So we have, these are the wells up here, and this is the ladder. And you take a ruler and you measure each of the fragments in millimeters from the well. So that one and that one and that one, each one of the fragments are measured, and then you create a graph of the distance. And now remember, we know the base pairs of each one of them, so we know how large the base pair size of each one of those fragments. So we create a graph of millimeters that they travel through the gel versus the base pairs, and you will get a graph like that. Okay, and then you take your fragments. So instead of guessing, trying to approximate, you go ahead and take your fragment, you measure the distance. So let's say, I don't know, let's say X. You come here, this is the distance X of your fragment, go up to the graph of the ladder, and then go across, and this is the number of base pairs in your fragment. That's the way to do it. We don't have that because we're working from a picture. It's a little hard to start taking a ruler and trying to measure millimeters from well to first fragment, well to second fragment, then create the graph. This is what's called a standard curve. Create a standard curve for the ladder, and then measure the distance between well and the first fragment, come to the standard curve for the ladder, and go across, and that gives you a much better approximation of the base pairs that you have. In our case, we're eyeing it. We're just saying, okay, this is between this fragment and that fragment. It looks like it's closer to this fragment right here. So it's approximately this many base pairs. Does that make sense?
Yeah, yes, I think so. It's just like the ladder shows like the distance between 23,000 and 9,416 base pairs was like pretty much the same as the distance between 6,557 base pairs and 4,361 base pairs. So it didn't seem like it was evenly, distributed between, it wasn't like if it's quarter, if it's 6 millimeters, it's going to be this many base pairs. So I was just wondering, about that.
Okay, so the distance travel didn't make sense in that, if I understand you correctly, in that something that's 20,000 base pairs was very close to something else that was 10,000 base pairs.
Yes.
Where you would think there would be more.
Yeah.
All right. And part of it may be that the gel might not have been run properly, maybe needed more time for things to separate more. That may be one reason.
So that was the picture. That, like there was like a little button
the picture that they gave us. The real deal. Yeah. Okay. I have to look at the picture to see exactly what the range of base pairs are, and then would go from there. But that's all we have to work with.
Yeah.
So as imperfect as it is, yeah, we're just gonna have to use that. And more than anything, I guess it's just make sure you understand the principle behind it. What is a ladder and how it can be helpful.
Okay.
Yeah.
I want to clarify.
Yeah, no problem. Okay. Any other questions? I want to just take a quick look at the let's see, at the lab report. This next PowerPoint is for chapter 10, so we'll do that one next week. But I do have the lab report, which is actually very, very short. And I do know, give me a second. Oh, there it is. Okay. The first part is just definitions. And then okay, let's see. Yeah, a lot of these we already did. What is the COM? You should know that from last lab report. Again, just review of how do you load the DNA, which side do you load it. The DNA fragments, the larger the fragment, the bigger it is, the harder it will be for it to run through the gel. The smaller the fragment, the easier, the faster it will go through the gel. So by looking at how far the fragment traveled from the well, you should have an idea of which is a bigger fragment, which is a smaller fragment. Yeah. Which is something we worked with last that report also, actually, the size of fragments versus how far they travel through the gel. Make sure you know the restriction enzyme, the temperature, the purpose of loading the diamond. Okay, we got that. The lithium bromide, just to give it some color. That's a intercalating agent. Controls. This is very straightforward. And then make sure you picture, and make sure it's a picture that we can see what's going on. Can see the samples. Which any of the patients? Okay, determine which one is, if any, are positive with the salmonella that's causing the outbreak. How many DNA fragments in the control sample versus in the patients? Okay, and then use the DNA ladder to determine the largest fragment in the control DNA sample. In page 4, which is the patient. Are you talking about question 4? 6 and 9. Okay, let's look. Okay, and then 9. Yeah, they are exactly the same question, actually. Salmonella match in the strain from the recent outbreak, and which of the patients showed to have the pathogenic salmonella? Yes, it's basically the same thing. Actually, it is the same thing. No, I'm not looking for, no, it's exactly the same. Worded a little bit different. And I can understand why I did it this way. As do I understand that the pathogenic salmonella is the one causing the outbreak and matching the control? And then what percentage of that would have been positive? Okay. Yeah, this is very straightforward. Any questions? Okay, very good. So if there are no questions, we'll meet again on Thursday to go over lecture. And I will post these. Will be a very oh yeah, go ahead. One question.
On the discussion board, how many questions are we supposed to answer per module? Is it per? I've been answering 2 questions per chapter. Pardon me?
Oh, no. It's 2 questions per module. Questions from one chapter, that would be enough. 2 questions per chapter is a bit too much. So
Yeah.
you've been doing a lot more than you needed to, but
Yes.
it's like an insurance policy. If one question wasn't correct, you had another one that was probably. So you were. But yeah, no, it's just 2 questions per module.
Oh, okay. All right. I was just thinking like 8 questions. This module is gonna be like
No.
video.
Nope. No.
For the night.
Okay, no problem. All right. Meet again on Thursday.
All right. See you then.
Okay, see you then. Bye-bye.